Thanks to everyone for joining on the early shift on the last day. So hardy souls you all are. We have presented inhaled oxytocin previously, so I'm only going to do a very sort of whistle-stop tour through the background and the history of the project so that I can get to the more recent data and the bioanalytical challenges that we've been facing. So postpartum hemorrhage is the leading cause of global maternal mortality, around 14 million cases per year. And it leads to between 50 and 80,000 deaths annually, depending on which estimates you use. And as with many of these global health issues, the burden of disease is highly inequitably distributed. So this is exemplified by the fact that in the UK, in the 3 years up to 2023, 14 women died of an obstetric hemorrhage, whereas in lower middle income countries, a woman is dying approximately every 6 minutes. And these are some of the countries that bear the greatest burden of that disease. And the tragedy of this, of course, is that most of these are preventable. Oxytocin is the gold standard therapy for both prevention and treatment of postpartum hemorrhage, administered as an IM or an IV injection. And consequently, the WHO recommends that every woman at every birth receives a prophylactic dose of oxytocin. The issue, though, is that the injection product is the only oxytocin product, and it requires refrigeration to maintain quality. And you can easily imagine that the cold chain infrastructure in lower middle income countries is not always complete. These products get exposed to ambient conditions in tropical climates. And as you can see from the graph on the right, which was a study we did with the UNFPA, deterioration can happen quite rapidly. So consequently, something like 40% of oxytocin in the field has been shown to be substandard, mostly due to that deterioration. In addition, as an injection, you need a skilled healthcare worker. In this context, it's a skilled birth attendant, and sterile needles and syringes to safely administer the product. So what we're trying to do as inhaled oxytocin is overcome these issues by having a non-injectable, heat-stable and convenient delivery system for oxytocin that can be accessed at all birthing settings. So where are we with that program? Well, we have a very robust heat-stable formulation. As a small peptide, we're using a technique which I'm sure many of you are familiar with. So we spray dry with in an amorphous matrix, trehalose with leucine as a shell former. And we have taken that initially for our initial clinical work. We took that spray dry construct, blended it with mannitol, magnesium stearate covered mannitol, just for manufacturability purposes so that we could put it into a capsule. And with that product, we now have a 3-year shelf life outside of the fridge and robust stability at higher temperatures. We've established a preclinical proof of principle in a postpartum sheep model by demonstrating comparability in terms of PK and PD to the standard of care, which is the intramuscular injection. And when we say pharmacodynamics here, we're talking about uterine contractility, because that's the mechanism of action by which oxytocin works and all uterotonics work to prevent postpartum hemorrhage. And then we took that capsule formulation with a rotahaler delivery system with GSK, as Michelle said, into the clinic. Now, before I look at that data, I think it's worth visiting our registration strategy. So what we are looking to do is register the product through the European Medicines Agency global health procedure, which is known as the EU Medicines for All procedure. It's targeted at products that are not going to be marketed in the EU, but they're targeting low and middle income countries. The EMA provides a stringent regulatory review in consultation with the WHO to provide contextual expertise. And then if you get a positive scientific opinion from that, you can get an expedited approval in low and middle income countries, mostly sub-Saharan African countries. So from that process, we've had the advice that we can register the product where we extrapolate efficacy from demonstrating pharmacokinetic comparability to the intramuscular injection, and then we generate a sufficiently large safety data cohort across the different birthing settings in which this product is targeted to be used. So with that in mind, our first in-human study was a simple ascending dose PK study. We looked at 4 doses from 50 micrograms up to 600 micrograms of the inhaled product compared to the intramuscular injection. And you can see that we got a nice dose-dependent PK profiles across the inhaled doses. But what was most encouraging in light of our registration strategy was the similarity between the 400 microgram oxytocin dose, inhaled oxytocin dose, and the intramuscular injection, which is the green and black profiles respectively here. So that's really kind of the update to what's been presented previously. From this point, we obviously wanted to and needed to demonstrate this same comparability in our target population. Our target population is women in the third stage of labour. The third stage of labour is the period between when the baby is born and the placenta is detached, and that's the time when oxytocin is indicated for prevention of postpartum hemorrhage. So we designed a study, you can see that here. Now, you can see in the study we had both non-pregnant volunteers, and I should have said that first study was in non-pregnant volunteers as well, and women in the third stage of labour. Just in the interest of time, I just want to concentrate on the third stage labour group here, which was a simple parallel comparison of that 400 microgram dose with the intramuscular injection. And we were targeting 20 women, and we had an interim halfway through with 5 subjects per group. What we discovered was what surprised and shocked us was that from both routes of administration, we barely saw any quantifiable concentrations in the plasma samples that we analysed. So you can just see down in the bottom corner here, if you see the marker here, these solid profiles here are what are the quantifiable concentrations from the TSL subjects. And you can see how much lower they are than the dotted profiles, which is the historical data from non-pregnant volunteers. In fact, we only had 4 subjects that had quantifiable samples. Now, we expected the concentrations to be lower because in pregnancy, volume of distribution is around 40% greater, but also, most importantly, we know that the protease enzymes that metabolize oxytocin ramp up at the end of pregnancy and throughout delivery. These are known as oxytocinase enzymes. It pretty much says what it does on the tin. But all the literature suggested we should still be in the quantifiable range. Now, our bioanalytical method is an LC-MS method with a lower limit of quantitation around 2 picograms per mil for this. So this was surprising to us. Our first instinct was perhaps we needed to revisit our sample handling. It was obviously critical for us to quash the enzyme activity in our samples post-sampling, and we'd done that using an EDTA tube in the original part of the study. But we'd come across another sampling tube from Becton Dickinson called the P100 tube, which had not only EDTA in, but a cocktail of proprietary protease inhibitors, which we thought might be able to add additional stability to the samples. And we analysed these both by looking both at oxytocin stability in ex vivo samples, but also the formation of known oxytocin metabolites. If we look at the oxytocin stability, you can see here that we spiked whole blood from women in late-stage pregnancy with oxytocin, and the stability across 90 minutes was similar in the EDTA tubes and the P100 tubes. Interestingly here, as a negative control, we also had heparin tubes that have no protease inhibition, and you can see just how quickly the oxytocin disappears if you don't have protease inhibition. But when we looked at the metabolite formulation, metabolite formation, this is a 7-amino acid metabolite of oxytocin, you could see its presence in the negative control, but we also saw the presence in the EDTA tubes, which suggests maybe we weren't getting complete enzyme inhibition in the original tubes. So with some other tweaks to the sample handling process, we restarted the study with these tubes. Unfortunately, no difference. So we had another interim at 7 subjects, and we saw similarly low concentrations of oxytocin. And because the study seemed to be futile from this point, we terminated at that point. At this point, we were fairly sure that our sample handling was robust, and we had to contemplate the fact that perhaps these were a real reflection of the physiological situation in the third stage of labour. But we wanted to interrogate this further, so we actually developed an assay for the oxytocinase directly. We did this by identifying an oxytocinase substrate, which is this LU-MCA compound at the top. I'm not going to say the whole name. But when it combines with oxytocinase, it hydrolyzes and releases this AMC moiety, which is fluorogenic. And we were able to show that that fluorescence, the rate of formation of that compound and the fluorescence that went with it was directly correlated with the enzymatic activity, and we validated that method. So with that method, we went back to our sampling tubes just again to reaffirm, were we getting oxytocinase inhibition in these tubes? And you can see it looked like we were in both of those tubes. But what surprised us was when we compared the oxytocinase activity in pregnant blood versus non-pregnant blood, and we saw this 11-fold higher rate of oxytocinase activity. Now, this type of study hadn't been done previously, but there were indications in the literature of what the increase in oxytocinase was estimated to be at the time of delivery, and it wasn't this high. And we begin to see that that combined with volume of distribution and other pregnancy effects likely was the reason that we were seeing such low concentrations. But then we asked the question, why was our data different to that was in the literature? And this came down to methodology. As I said, we had an LCMS method. Most of the PK work that's been done before on the injection, because the product's so old, was pre-1990s and used ligand binding assays. This is just one profile here where you can see plasma concentrations in the tens of picograms per mil, which would have been readily quantifiable by our method if we'd seen that. But what is emerging is evidence that these methods are actually unreliable because they're not specific to oxytocin and they cross-react with some of the oxytocin metabolites. They're also very sensitive to how you prepare the sample. So we wanted to look at this. So we validated a commercially available oxytocin ELISA assay, and we looked back at our clinical samples. Now, of course, we only have 1 or 2 profiles from third stage labor that had any meaningful oxytocin concentrations. So we were limited in what we could do, but this is just one subject on the left here. And you can see that when we reanalyzed this sample, almost by an order of magnitude, the apparent oxytocin levels as measured by the ELISA were higher than those in for the LCMS method. On the right is non-pregnant blood. Now remember, this blood has much lower quantities of oxytocinase activity. And so you can see the concentrations actually mirrored each other quite nicely until sort of 10, 20 minutes, and then they began to diverge. And that divergence probably speaks to the small amount of oxytocinase metabolism that's happening on the oxytocin. It continues to be picked up in the metabolites by the ELISA, but we're not seeing it in the LCMS method. So this was all great. This was, wonderful scientific stuff that we could publish, and, I'm in an academic institution. This is awesome. Except this placed our registration strategy in tatters unless we could develop a new bioanalytical method that was sufficiently sensitive for us to characterize PK effectively in the third stage of labor. We already had a really sensitive method at 2 picograms per mil. I was somewhat skeptical, as were some of the others, but at this time we were partnered with J&J. They were up for the challenge, and lo and behold, they were able to be successful. So they developed a new method using an ultra-sensitive piece of equipment, the nano LCMS, and they combined it with an immunoprecipitation method of sample preparation using antibody beads specific to oxytocin. And as you can see here, they were able to validate that method down to 100 femtograms per mil, which I think is quite remarkable. This is a 3-year piece of work that I've just described, but we still had some residual samples from the original clinical study, 5 subjects from each dosage route of administration. When we reanalyzed the samples, lo and behold, we could see fully characterized PK profiles. The variability was a bit higher than we were used to, but what was really encouraging was once again, we were seeing this comparability between the intramuscular and the inhaled. So we were back on track in terms of our registration strategy. So, and just to re-emphasize the point about the difference between the non-pregnant and third stage labor, this is the inhaled dose with the characterized profile of the third stage labor patients with the non-pregnant. So you can see it's a marked difference. So just to finish, I just want to bring an update on where we are with the program. In parallel to some of this work, what we were looking to do also was to find a new device for the program. The rotahaler was great as a clinical prototype, but it's not appropriate for low and middle income use. Separate capsules is difficult when you're in a low resource birthing situation, and the device, to be blunt, was too expensive, even though it is still simple. We did an extensive review, and we settled on the Iconovo ICO-1 device, which you can see samples of in the exhibition, and it pretty much ticked all of our boxes of being ultra low cost, simple to use, and self-contained. But obviously, we needed to demonstrate that same similarity to the intramuscular injection with this device as we'd shown previously. So we took this into another phase 1 study, going back into non-pregnant female volunteers, and we added some extra elements here. So in the first 2 dosing sessions, what we were looking at was comparing 2 doses of oxytocin, 150 micrograms and 600, with the injectable product. Our PK modeling had suggested that those 2 doses would sort of equally bracket the IM exposure, and that's what we were hoping to see. The intravenous dose, which I haven't shown you previously, is there as a safety reference. We know that intravenous administration of oxytocin can lead to some cardiovascular side effects, so we wanted to make sure our PK didn't approach those levels from the inhaled dose. And then on the dosing session 3, we had 2 objectives. The first one was to give an ultra high dose of the inhaled, which is this 3 times 300 microgram dose, so a 900 microgram dose, to give ourselves an upper safety reference that we hopefully would show no side effects. And then we did a repeat dosing schedule with those 3 doses, but at 10-minute intervals, and that replicates treatment use of oxytocin. So if a woman does continue to bleed, even after prophylaxis, you can give repeat doses subsequently to try and achieve cessation of bleeding. So this data that I'm about to show you is IMPRESS. I know this is a futile, but if you could not distribute it any more widely, that would be great, because we're expecting publication in the next few weeks. We're really happy with the data that we saw here, and we felt that we achieved our objectives. The 2 inhaled doses on the left-hand side, you can see bracket the intramuscular and are similar in terms of characteristics, so we still saw that comparability that we were seeking. On the right-hand side, you can see the black profile is the interval dosing, and the blue profile is 900. The key part about this, apart from just characterizing them, was that we saw no substantial side effects on either of those profiles or across the whole study as well. So we think this puts us in a good position in terms of having a product and a device to go forward. So that leads me to talking about, just in the last couple of minutes I have, how do we go forward? Well, and where are we on this? Well, on the plus side, we think we have a strong product with really robust technical data to support it. We're back on track for that streamlined regulatory pathway, and we have a product that's been independently assessed on numerous occasions that's having the potential to have huge impact if we can deliver this into the right settings. On the minus side, and it's a fairly big minus, is that we have little to no funding or partners to take this forward anymore. So what we need, obviously, is to develop a commercial manufacturing line, then to do our pivotal clinical trials and take it forward. It's not a difficult development program, and in the context of commercial pharmaceutical development, it's a pretty low-cost development program. But unfortunately, our barrier to investment is that nobody can make any money from this. So because this product is targeted in low and middle-income countries, there is no potential for commercial return. So we're looking at a philanthropic play. But you might say, well, you know, women give birth in high-income countries, surely they would use this product as well. Well, the reality is actually very few women give birth in high-income countries. There's 142 million births a year. Fewer than 10% of those happen in high-income countries. So, you know, that's 10 million births. So even if we were used in every birth, that's only 10 million units per year. And we would only get a small segment of that because the injection is very readily and easily used because we have trained healthcare workers and cold chain infrastructure and so forth. So we're looking at this commercial barrier to impact. So my plea to you today is, how can we get together and get everyone together to help us do this? Because we believe the only way we're going to do this is to bring together a coalition of the willing, probably across both public, philanthropic, private sectors, so that we can de-risk it for everybody and bring in an industry partner that can take it forward. We believe that once the development is done, we can supply this sustainably at a cost that will cover manufacture cost and distribution and be affordable by the national governments who would be our procurers in these countries. But the barrier is this development cost there is to go forward that we need to overcome. So please, if you can help us, come and talk to us. Talk to myself or Jacob Harker and Eddie French, both of whom I'm sure somewhere in the audience here, or speak to your networks, and let's see if we can do this together and have a real impact for these women that are dying needlessly in so many countries. Thank you.
Thanks, Pete. It's fascinating. It's sort of, how can I say, encouraging, but also heartbreaking that you've really navigated some of the technical challenges that you've had, but you've still got these hurdles with investment just from a commercializable perspective. And I really wish you every success with getting additional support. Do we have any questions from the audience to Pete? If so, please put your hand up, or feel free to use Slido if you're not feeling, or you don't have, a bit like me, don't have much voice this morning. Can we see anyone? Okay. Well, oh, we have somebody over there. Thank you.
Thank you for presenting such a very interesting study, as well as the intention behind that. So thanks for that.
Thank you.
I just have a quick question concerning deciding upon the dose, because the intravenous injection data was already available, and when first time you wanted to go inhalation route, I'm just wondering how you decided what dose you should be starting with.
Yeah, so the dose ranging that we did in the preclinical sheep model was the first indicator of where we should start with that, and that was what drove that 50 to 600 microgram dose range that we did in the first clinical study. It's interesting because the registration strategy, we talk about comparability with the intramuscular injection, but you may have picked up that in the WHO guidelines, you can give the same dose of oxytocin either intramuscularly or intravenously is the recommended dose. But you will have noted from the PK profiles that there's quite a difference in exposure between the intramuscular and intravenous, as you would expect. So actually, we believe we have quite a large target dose window of just achieving a pharmacokinetic profile that's at least as good as the intramuscular, but stays in that window below the intravenous, which actually is quite a wide gap. But yeah, it was an iterative process from the sheep studies initially, and then this study. And what I should have mentioned there is we've modeled the data that we've had in the final study, and we see that the IHO-1 product that we've developed is probably 20-25% more efficient than the Rotahala. So the likely comparable dose to the intramuscular is around 300 micrograms, not 400 micrograms. But obviously, we need to prove that going forward once we have a product that comes off a representative manufacturing line.
Thank you.
Do we have any more questions from the audience? Okay, we have one from Slido, and it's basically asking, when developing a bioanalytical method, should you be using whole blood samples or plasma samples if you're developing and validating these LCMS methods for analyte detection?
Okay.
Quite a technical question there.
Yeah, absolutely. And I am not a bioanalyst, so I'm going to try and answer this. What actually was really interesting, and in the publication that's cited when we were talking about the ex vivo metabolism, was what we found was that if you add EDTA to whole blood, you get complete suppression of the oxytocinase. If you add it just to the plasma post centrifugation and so forth, you don't get the same suppression. So we did look at this, but I'm not going to say anything further than that because it's beyond my area of expertise. But I'd point you to the papers, and it talks a lot more about how we developed the method and what's appropriate for that. So,
thank you. Time for one last question? Oh, over there. Thank you.
And before David speaks, I just have to point out he is a co-inventor of the technology and has a long history with this project as well.
Thanks, Pete. No, it's great to see you. I mean, it's sort of a real mixed blessing, the whole situation. Question really following on from the innovation session yesterday, and how is, in all this exploration for funding, how do you see the challenges? I mean, you've got some really big names of funding partners there.
Yep.
There are an amazing number of philanthropic bodies. What have you learned so far about how to get to the core of finding, the net worth, the sort of the large philanthropic funding bodies? What's your experience?
I think the key takeaway for me, not just in terms of the philanthropic funders, but also the pharma companies, is it comes very much down to individuals. Their individuals hold a lot of power in these situations. So, for example, we used to be funded by Gates. We had a champion internally. When they left, then our funding stopped. The interest from J&J, and hopefully I'm not speaking out of turn here, and it's our collaborations with both were fabulous, and they took us an awful long way through this. I didn't talk about inhalation tox that we'd done with GSK and so forth. But it's very much comes from the leadership. So when Andrew Whitty was CEO of GSK, there was a strong focus on global health. When he left, that minimized. Similarly, Paul Stoffels was the chief scientific officer of J&J, and they had a big program. When he left, it was minimized and there was a focus back on commercial products. And that's their choice, right? They're commercial organizations. But in terms of the philanthropic, it's really the big organizations, it really depends on who's in place in the different sections of the organization as to whether you get traction. The other thing is, as you said, there's a lot of philanthropists out there that don't advertise themselves for obvious reasons. You know, there's not websites for every foundation, those kind of things. So being able to just get into that network of philanthropy and getting some champions on your side. So for example, just as a shout out to the Planet Wheeler Foundation, which actually no longer exists, but the trustees of that foundation, which is a small value foundation, have been huge advocates for us in the philanthropy networks, talking to high net worth individuals. So I think that's just some of my immediate reflections, David. Yeah.
Okay, thank you. In the interest of time, we'll move on, but thanks very much, Pete. Really fascinating talk.